Miscellaneous compound. Pyriproxyfen butoxide, or PBO, is a methyl-dioxyphenyl chemical that is utilized in the management of arthropod pests as a synergistic additive. It is used in conjunction with pyrotherins as a synergistic agent. With this, the effect of insect growth regulators is also amplified. Arthropods' microsomal enzyme system is inhibited by this chemical. It has minimal toxicity to mammals and degrades quickly in the environment. Pyrotherins, jasmolines, citronella, indolone, garlic oil, DEET or N-diethyl-M-toluamide, and DMP or dimethylphthalate are among the compounds used to repel insects. Pyrotherins or pyrethrum is a natural active ingredient of plant origin used in veterinary medicine in dogs, cats, and livestock against some external parasites such as lice, fleas, flies, and etc. They are also used against agricultural and household pests. They are found in some plants such as Chrysanthemum cinerariae folium, Chrysanthemum coccineum, Tagetes minuta, or wild marigold or black mint, Tagetes erecta, or Mexican marigold or Aztec marigold. The mechanism of action of pyrotherins are similar with synthetic pyrethroids. It acts on the membrane of nerve cells, blocking the closure of the ion gates of the sodium channel during repolarization, which strongly disrupts the transmission of nervous impulses. At low concentrations, insects suffer from hyperactivity, and at high concentrations, they are paralyzed and die. Citronella is found in citrus such as orange, lemon, particularly in Citrus hystrix. Its mechanism of action is that it directly activates calcium channels in the neurons of several mosquito species in a similar way as the overexciting and repellent effect of pyrotherins. In Drosophila melanogaster flies, citronella is detected by protein receptors known as TRP or transient receptor potential channels. Once activated, these receptors send signals to the insect's brain that cause an aversive response. DEET or N-diethylmetatoluamide remains the most effective among currently available insect repellents for humans. It is a broad-spectrum repellent that is effective against mosquitoes, biting flies, chiggers, fleas, and ticks. However, the effectiveness of DEET formulations for dogs and cats has not been proved, and safety is a concern because concentrated formulations containing DEET have caused weakness, paralysis, liver disease, and seizures in pets. Insect growth regulators. There are a new category of insect control agents which constitute a chemical group that interferes with growth and development rather than killing the target. It acts primarily on immature stages and not employed in adult control. IGRs have an effect on specific hormones in insects, which humans lack. Despite the fact that they do not kill insects instantly, they can prevent a pest population from reproducing until all of the bugs have been eliminated. How do insect growth regulators work? In adults, generally, they survive IGR therapy and continue to be a nuisance until they naturally die. However, the eggs they lay may be infertile. Adults' reproductive organs can occasionally be impacted, rendering the adult sterile. In eggs, IGR-treated eggs may never hatch. Even if the egg hatches, the young insect may die. In larvae, after exposure to IGRs, the larva or worm-like stage may be unable to develop normally into an adult. Certain larvae may remain at this juvenile stage until death. In pupa, Typically, a cocoon similar to a moth's cocoon protects the pupa. IGR treatment may prevent the pupa from maturing into an adult and reproducing. Insect growth regulators is based on the mode of action, such as chitin synthesis inhibitors, chitin inhibitors, and juvenile hormone analog. An example of chitin synthesis inhibitor is benzoyl phenylurea. It can kill young insect stages that are exposed to them. Insects can't complete ecdysis and die during the molting process. During each molt, chitin is synthesized by polymerizing individual sugar molecules. This is combined with proteins to form chains, which are then organized into microfibrils. Although the exact mechanism of action is uncertain, it may include interfering with the assembling of chitin chains into microfibrils. In addition, BPU exhibits a transovarial effect, which means when adults exposed to the chemical produce eggs, the chemical is ingested into the egg's nutrition. Therefore, the development of the egg is still normal. However, the newly hatched larvae are incapacitated to hatch. BPU is effective against insects but not ticks, with the exception of fluazeron, which is effective against ticks and mites. BPU is highly lipophilic, which means it accumulates in body fat before being released into the bloodstream and eliminated unmodified. An example of benzoyl phenylurea are diflubenzuron and flufenoxaron, also Leuphanerone. Diflubenzuron provides protection for 12 to 14 weeks. It is more efficient against first-stage larvae than second and third instars and is therefore recommended as a preventive. Topically administered diflubenzuron is poorly absorbed through the skin. After oral administration to cattle and pig, about 70% to 85% of the administered dose is excreted and changed in the feces, and about 5% in urine. In sheep, excretion through urine and feces is equally distributed. The mechanism of action of diflubenzuron is that it hampers the synthesis or the correct deposit of chitin in the cuticle of insects. As a consequence, larvae or nymphs cannot properly molt and die during the molting process. Egg hatching is also interrupted due to the fact that young larvae developing inside the egg have to molt before hatching. If the adult female was treated with chitin synthesis inhibitor, significant amounts of it are passed to the eggs. The embryo can develop quite normally, but it dies during the first molt still inside the eggshell. So this is the dosing recommendations of diflubenzuron. Leuphanerone is administered orally. It accumulates in fat and is slowly released. Fleas ingest it with their blood and pass it to eggs that are no longer visible after 24 hours. Here's the dosing recommendations of Leuphanerone. Fluazeron is a specific tick development inhibit or with systemic effect. Abundantly used in cattle, mostly in pour-ons. Fluazeron is available in some countries for use in cattle as a tick development inhibitor. When applied as a pour-on, it provides long-term protection against the one-host tick Boophilus microplus. Fluazeron has a systemic mode of action. After topical pour-on administration, it is absorbed into blood either through the skin or after licking. Once in the blood, it is distributed throughout the whole body and is deposited in the fat tissues. This deposit is slowly released back to the bloodstream and reaches the tick everywhere in the host's body. This allows maintaining the effective concentration in blood for up to 3 months. It is hardly metabolized. It is excreted slowly with a half-time of about 13 days, mainly through feces. Here's an example of dosing recommendations for fluazeron. Another mode of action of insect growth regulators is the chitin inhibitor. Thiazine or pyrimidine derivatives, chemically distinct from BPU in structure and manner of action. It inhibits the deposition of chitin into cuticles. Cyromazine larvidextrazine derivative. When larva 1 comes into touch with another larvae, the molts to the second and third instars are hampered. Pour-on preparation is effective for 13 weeks after a single pour-on. The dips or shower impact is more effective for a longer period of time. Dicyclonyl is an example of a thiazine pyrimidine derivative that, when applied topically to diptyn larvae as a pour-on preparation, provided protection for 20 weeks. It is applied in the control of blowflies. Blowflies, houseflies, and mosquitoes are also susceptible to this insecticide. The mechanism of action of dicyclonyl has not been elucidated. It is certainly not a chitin synthesis inhibitor like other insect development inhibitors such as the benzolurea. It seems to act upon the molting process in a similar way as cyromazine, probably interfering with the process of chitin deposition. Topically administered dicyclonyl is poorly absorbed through the skin of sheep, at least 2% to 4%. The absorbed dicyclonyl is partly metabolized and excreted through both urine and feces. About 95% of the administered dose is excreted in the 7 days following treatment. So here is the dosing recommendations for dicyclonyl. Cyromazine, a thiazine pyrimidine derivative, is quite specific and effective against dipteran larvae, but ineffective against other external parasites such as lice, mites, ticks, etc. It is also abundantly used for off-animal treatment of livestock facilities against any kind of flies, and it is not used in pets. At recommended dose rates, cyromazine shows only limited activity against established strikes and must therefore be used preventively. Here is an example of dosing recommendations for cyromazine. Another mode of action of IGR is the juvenile hormone analog. This prevents metamorphosis to adults by mimicking the activities of natural juvenile hormone. Enzymes in the insect's circulatory system eliminate the endogenous insect hormone after the larvae are fully matured, allowing them to develop to adult stage. Unlike the estrase, the juvenile hormone analog does not destroy itself by binding to the JH receptor site. As a result, the process of maturation and growth to adulthood is slowed. Here are some JHA examples: Methoprene, Pyriproxyfen, and phenoxycarb. These compounds also have ovicidal and embryocidal activity against flea eggs when applied topically to dogs and cats. Female fleas in the hair coat absorb the juvenile hormone analog, which affects viability of developing eggs. Methoprene is a terpenoid compound with a very low mammalian toxicity that mimics a juvenile insect hormone and is used as a feed-throw larvicide for hornfly control on cattle. Here's the dosing recommendations for Methoprene. Methoprene and other JHA suppress or stimulate the expression of various genes involved in insect metamorphosis, otherwise regulated by natural juvenile hormone. Depending on which gene is affected, different biochemical and cellular effects will result. The bottom line is that development is disturbed and interrupted. So this picture shows the effects of the juvenile hormone analog insecticide, Methoprene, on mosquito development. Another juvenile hormone analog is the Pyriproxyfen. In rats, Pyriproxyfen concentrations were found to be highest in the fat. Without evidence of accumulation, Pyriproxyfen was highly and rapidly metabolized and excreted into feces and urine, with the major route of excretion being via the feces. Here's the dosing recommendations for Pyriproxyfen.